Hey everybody, welcome back to General Biochemistry Lecture. This time we're moving on to Chapter 15, talking about the metabolism of glycogen in animals. Here's the key principles for this chapter. It's a much shorter chapter than Chapter 14, but it is equally important. We're going to start with talking about the structure and function of glycogen. Now we touched on glycogen already, that it's a storage form of glucose, primarily found in the muscle and the liver. When you break down glycogen in the muscle, you're delivering glucose so that you can actually, contract your muscles. So if you're doing some kind of vigorous workout or you need to run from a bear, then you can provide an extra boost of energy. The glycogen stored in the liver is kind of a reservoir that's used to maintain blood sugar, blood glucose. So here's some imaging of glycogen beta granules. So these are cytosolic granules. They vary in size and structure and localization. So pretty much they can do what they want, right? But you can see these very dense particles. Those are the glycogen beta granules. Alpha granules are just protein-rich granules that have a bunch of beta granules. They release glucose slower than beta granules, and they're often associated with the tubules of smooth ER. So what's the structure of glycogen? Let's look at a beta granule. At the center, you have glycogen as a dimer, and we're going to talk about this protein later on. But for right now, we're just going to talk about it very casually. This dimer serves as a primer for the growing glycogen chain. Then you've got tiers of glucose residues that are in alpha-1,4 linkages that are linked with alpha-1,6 branches. So there's lots of free non-reducing ends. You can see kind of the different tiers of the glycogen. We're looking at all of the branching. It's a very, very big, very big structure. Let's talk about how we break down glycogen and how we rebuild our stores. The two processes for that are glycogenolysis, which is the breakdown, so lysis is splitting, and the result of that is glucose 1-phosphate. Glycogenesis is you're synthesizing it, so genesis, making something, synthesizing it. Glycogen breakdown is catalyzed by glycogen phosphorylase. Remember the difference between a kinase and a phosphorylase. We're breaking a bond and then adding a phosphate to replace that bond. It catalyzes the phosphorylated cleavage at the non-reducing end of the glycogen chain. You have to use pyridoxal phosphate, and this enzyme is just going to keep chewing away at that glycogen until it reaches a point that is 4 residues away from an alpha-1,6 glycosidic linkage. Once we get there, we need a different enzyme, and that's the debranching enzyme. What it does is it rearranges the glycogen chain so that the shorter chain gets incorporated into the main chain. So you lose that branching. So first you use the glycogen phosphorylase, chew until you get to a place where you're really close to that linkage, that branching point. Then The transferase activity of the debranching enzyme comes in and says, don't worry, I'll save the day. And then it creates a longer chain for the glycogen phosphorylase to eventually come back in. But what it also does after it debranches is it gets rid of that glucose that has the alpha-1,6 glycosidic linkage. Once that happens, then you can go back to using the glycogen phosphorylase to continue liberating glucose units one at a time. So we said that this process yields glucose 1-phosphate, and that isn't helpful for what we need to do if we need to do glycolysis. If we need to do glycolysis, then we have to use phosphoglucomutase, and that catalyzes the reversible conversion of glucose 1-phosphate to glucose 6-phosphate. This mechanism is something that you should be able to draw. It's very similar to other group transfer reactions that we've seen, particularly the mutase in glycolysis, where we're doing the same thing only with 2 histidine residues versus a serine, which is shown here. Serine is already preloaded with a phosphate. It attacks the C6 position. Then you have the phosphate on the C1 position, able to attack the serine and replenish that phosphate group on the serine. And then that's it. It leaves. So with glucose 6-phosphate, there are different things that can happen, right? We talked about the pentose phosphate pathway in chapter 14. There's also glycolysis. In the skeletal muscle, glucose 6-phosphate enters glycolysis. In the liver, there are some different options. So there's glucose 6-phosphatase, which can take that glucose 6-phosphate and chop off the phosphate to make just plain old glucose that can then be exported to the blood to replenish your blood glucose. So let's talk about that. In the cytosol, you've got your glucose 6-phosphate. There's a specific transporter that allows that molecule to enter the ER lumen. So this transporter is on the membrane of the ER. There's a glucose 6-phosphatase that is also embedded in the membrane. The active site is located in the lumen, not in the cytosol. And that's to keep this reaction separate, away from all the other reactions that are going on with glucose 6-phosphate. That's going to, again, chop off that phosphate group and yield free glucose and inorganic phosphate. There's another transporter, a glucose transporter, that will shuttle the glucose out of the ER lumen. We do the same thing with the inorganic phosphate. There's a transporter. And that glucose is then shuttled outside of the cell, where it can go into the capillary and increase the blood glucose concentration. So this is a process that you will need to understand. And you don't need to go in any more in depth than what I've shown you here and what I've talked you through. So this only happens in the liver, because the liver is responsible for maintaining our blood sugar. The liver and the pancreas together, really. So just like anything else, if you have a major metabolic pathway, there are diseases if there are genetic defects. So here's a whole list of glycogen storage diseases. We're not going to go through all of these. You're not responsible for knowing the different types and the names and all of that. But if you're interested, the world's your oyster. Let's talk about some other ways that we use glucose. So we can take glucose and add a UDP, and that is how we actually make glycogen. So the sugar nucleotides, you've got an anomeric carbon of a sugar attached to a nucleotide. We need to do this reaction so that we can form UDP and activate the glucose to add it to a growing glycogen chain. So let's say that we've got a sugar with a phosphate on it, and we have some type of nucleotide. What we're going to do is the phosphate, there's one of the oxygens on the phosphate is going to attack here. And you're going to liberate pyrophosphate, and you're going to make a sugar nucleotide. So that's a diphosphate, because remember we've got two phosphates. You've got your ribose and then the base, so A, U, G, whatever it is. That pyrophosphate, which is just two inorganic phosphates still bound together, is going to get chopped up into two inorganic phosphate molecules. And this reaction has a large negative delta G. So that reaction helps to drive the formation of the sugar nucleotide. Sugar nucleotides are great for biosynthetic reactions. Their formation is pretty much irreversible. That large negative delta G of breaking up that pyrophosphate, you're not undoing that. So it is irreversible. The nucleotide moiety has a lot of little groups that can interact with an enzyme. So it gives the enzyme more surface so that it can create specificity for a particular substrate. The nucleotidal group, so UDP, ADP, they're great leaving groups. So you can use these molecules to facilitate nucleophilic attack if you want to add a sugar to something else. And tagging with the nucleotidal group will distinguish whatever sugar from the other pool of sugars, so that you know, okay, this sugar over here with the the ADP or the UDP or whatever it is, that's going to be used for this process over here, so leave it alone. So it's a great way to distinguish pools within your sugars. So let's begin the process of glycogen synthesis. You take glucose 6-phosphate, which can be from one of the hexokinase isozymes. Remember, that takes glucose and slaps on a phosphate group. It can also be from lactate. So remember that lactate can be used to form pyruvate and then go through gluconeogenesis to create glucose 6-phosphate. Between those couple of sources, you've got a good pool of glucose 6-phosphate. You can also have the phosphoglucomutase that we talked about, that converts glucose 6-phosphate to glucose 1-phosphate. Well, it can do the reverse, right? It's reversible. So you can take the glucose 1-phosphate from glycogen and convert it to glucose 6-phosphate. One of the key steps is the UDP glucose pyrophosphorylase. It takes glucose 1-phosphate and converts it to UDP glucose. So for this process, we're taking that glucose 6-phosphate and converting it to glucose 1-phosphate using the same enzyme that converts the 1 to the 6. So just to clarify. Now we're going to take the glucose 1-phosphate from the result of the mutase reaction, and we're going to add on a UDP and release that pyrophosphate, which again gets broken up to make two inorganic phosphate molecules. After that, you're going to add on that glucose to the non-reducing end of glycogen. I know we talk about, oh, it's not the non-reducing end, so chemistry can't happen there. Yes, it can, just not that kind of chemistry. So we need glycogen synthase. It transfers the glucose from the UDP glucose to the non-reducing end of a branched glycogen molecule, and it forms that alpha 1,4 linkage. The mechanism is shown here. You've got the non-reducing end of a glycogen chain, but there is still a hydroxyl group here, and that oxygen can actually attack the phosphorus in the pyrophosphate, so the DP part of the UDP. You release UDP, it's a great leaving group, and you form a link between the glucose that was on the UDP molecule and the glycogen chain that already exists. If we need to branch, there's a glycogen branching enzyme. Just like there's a debranching enzyme, we have a branching enzyme that's going to catalyze the formation of the alpha 1,6 bonds at the branch points. So glycogen synthase is great, but it needs a primer. It can't just start, adding on glucose to nothing. It needs a little starter. And that, we're going to go back to our friend glycogen, which is at the core of these granules. Glycogen has the primer that you make new chains of glycogen on. It also has the enzyme that catalyzes the creation of the primer. There are several important residues here. Tyrosine 194 and aspartate 162. This enzyme actually uses manganese, not magnesium. And that manganese helps to coordinate the UDP glucose, which is bound here. Remember that this is a dimer. So this is monomer 1, this is monomer 2. So the first reaction, we have the autocatalytic formation of a glycosidic bond between the glucose within the UDP glucose and tyrosine 194 of glycogen. Because there's two, we have a dimer, so one can help to add that glucose onto the other. And that reaction is shown here. So we've got tyrosine that is part of the active site. It's got a hydroxyl group, suspiciously like the non-reducing end of a glycogen chain. That's going to attack the anomeric carbon on the UDP glucose. The leaving group is UDP. And then we have an attachment to this tyrosine. Once we have that, then we can have the reaction happening again and again, only this time it's the hydroxyl on the non-reducing end of the glucose that again attacks the anomeric carbon of UDP glucose, and that is how we create a small primer that can be acted on by glycogen synthase. Now there is an aspartate residue in the active site that is thought to be part of this mechanism as well, to help aid in properly orienting the sugar on that tyrosine. Now we're going to look at the regulation of the breakdown and synthesis of glycogen. These two processes are complete opposites, just like glycolysis and gluconeogenesis. So you don't want them both running at the same time. Glycogen phosphorylase in the skeletal muscle has two different forms. This is also true for the liver. There's glycogen phosphorylase A, and that one is active. So A, active. Glycogen phosphorylase B, much less active. We didn't say completely inactive, but it is much, much less active than the A form. Epinephrine, which gets released from vigorous muscle activity, and glucagon, which acts on the liver, trigger phosphorylation of phosphorylase B, converting it to phosphorylase A. So phosphorylation, there's a couple of serine residues that get phosphorylated and activate the phosphorylase. The phosphorylation event coincides with some conformational changes that allow for increased activity. We're going to talk about all the regulation that you see in this figure so that you can actually explain how glucagon and epinephrine signal to create these changes for phosphorylase. So first we're going to talk about the overall cascade. Right? Now we didn't cover enzyme cascades. There is a chapter that we did not cover, but we're going to talk about it here. I figured that it would just make sense to talk about it in the scheme of a metabolic pathway versus isolated on its own. So we're going to talk about this process generically first, and then we'll talk about the specific differences between what happens in the muscle and what happens in the liver. So you have the binding of an extracellular signal, and that causes a cascade. What happens is the signal binds on the outside, and it activates a trimeric protein that creates a signal on the inside of the cell that activates a cascade of enzyme activity. So we've got the stimulation of the alpha subunit of the heterotrimeric G protein, which there are lots of these proteins, and there are ones specific to each of the different extracellular signals. So once you stimulate that G alpha, then you're going to stimulate adenylyl cyclase, and that's going to create an internal signal of cyclic AMP, which is also abbreviated with the little c in front of AMP. So you make some molecules of that. Eventually that signal gets turned off, but that cyclic AMP goes on and activates protein kinase A, or pKa. Once that enzyme is active, it can go and phosphorylate the phosphorylase B, or kinase. And that phosphorylase B kinase becomes active when it's phosphorylated. Now we've got active phosphorylase B kinase. You can probably guess what that's going to do. It is going to phosphorylate glycogen phosphorylase B. Which, by the way, you'll see this referred to as phosphorylase or glycogen phosphorylase. That's because in the literature you see it written as phosphorylase is the first one that was discovered. So people just kind of do that. Strange aside. Anywho, once you have active glycogen phosphorylase, it is now in the A form, and it can do some work. So it can take that glycogen and start chewing it up to make glucose 1-phosphate. You'll notice that the numbers along the side here, 20x, 10x, 100x, what this is showing is the amplification of the signal. To go from a few molecules on the outside of the cell to thousands of glucose 1-phosphate molecules, which can then have their whatever fate depending on which tissue you're in. That is huge amplification. Now we're going to go through and talk about some of the differences with the muscle and the liver. So we'll start with the muscle. So myocytes, those are muscle cells. Calcium ions are a signal for muscle contraction. Calcium ions bind and activate phosphorylase B kinase. It has a calmodulin domain to it, and it is activated by calcium. So an increase in calcium also occurs along with this signal, epinephrine, to activate this phosphorylase B kinase. So that's one difference. And again, we're talking about muscle. AMP accumulates when you're doing some kind of vigorous activity. And that AMP binds and activates the phosphorylase. So that's going to speed up the formation of glucose 1-phosphate release from glycogen. ATP blocks AMP binding, so that will inactivate the enzyme. Which makes sense. If we already have enough energy, then there's no need to keep doing this process and releasing more glucose. That glucose 1-phosphate gets converted to glucose 6-phosphate, and then it goes to glycolysis, which helps to fuel muscle contractions. So it helps to maintain the muscle contractions because you're making energy. Once that happens and you need to turn the signal off, then we go to a protein called phosphoprotein phosphatase 1, PP1. And what it does is it removes the phosphoryl groups from those serine residues on the phosphorylase A and returns it to the less active form, phosphorylase B. So that turns off the activity. Now let's talk about the liver. In the liver, instead of epinephrine, we're talking about glucagon. Glucose is gone. We need to increase the blood glucose level. Glucagon binds. We have the same cascade. There's no calcium involved here. That's in the muscle. So we just have the straight up signal amplification. And then once we make all that glucose 1-phosphate, we eventually end up making glucose. So we need to cut off that phosphate group with a phosphatase. And then we need to shuttle all of that glucose out into the blood to raise the blood sugar. Once you have an adequate blood sugar level, that glucose is going to start re-entering the cells in the liver, the hepatocytes. And the binding of glucose inactivates the phosphorylase A. Glucose binding makes it so one, it binds to an allosteric site, which means that it's a site that's far away from the active site. And what it does is it makes those phosphorylated serines stick out like sore thumbs so that they can be cleaved. And then insulin will also signal to PP1, hey, you should go and turn off that phosphorylase A because we've got plenty of glucose in the blood. So when you have phosphorylase B in the liver, it will also be bound to glucose. So glycogen synthase is also subject to lots of regulation. Glycogen synthase A, unphosphorylated, catalytically active. Glycogen synthase B is phosphorylated and inactive. So the A form is active, the B form is inactive. And it's the same thing for both of these enzymes. So we just talked about how we're chewing up the glycogen. Now we're talking about how we make glycogen. And phosphorylation is the key to all of these things. There's also glycogen synthase kinase 3, which is responsible for phosphorylating glycogen synthase A. So that is the enzyme that will turn off glycogen synthesis. So let's talk about how this is regulated. Insulin inactivates the kinase, so GSK3, that glycogen synthase kinase 3, and it activates PP1. We already know that, the second part about activating PP1. So activating PP1 means that you're going to turn off glycogen breakdown. So insulin says, stop breaking down glycogen. And I also need you, GSK3, to stop phosphorylating my glycogen synthase because I need it to be active. Once you inactivate GSK3, that means that PP1 can also go and dephosphorylate glycogen synthase A. So this process turns on glycogen synthesis. Glucose 6-phosphate acts allosterically to make glycogen synthase B a better substrate for the phosphatase. So when that is bound, it says, hey, you see these phosphate groups? Please take them off. So as you're thinking forward to assignments and exams and being able to kind of pull everything together, you really want to think about insulin and glucagon signaling and what that does to the pathways that we've covered in this chapter. So glycogen synthesis and glycogen breakdown, and then also relate that to chapter 14, what it does to glycolysis, gluconeogenesis, and the pentose phosphate pathway. So let's talk more about GSK3. It is hierarchical, and what that means is it doesn't just act on its own. It can't phosphorylate glycogen synthase until another kinase comes in and phosphorylates it first. So that's called a priming event. And the kinase in question is casein kinase 2. So it comes in, casein kinase 2, and it phosphorylates a priming site near the cysteine that needs to be phosphorylated by GSK3. Notice that there are several residues positioned to interact with that phosphate group on that serine that is close to the other sites. So think about why those residues can interact with that phosphate group. What is it about their R groups that makes them good to interact with that phosphate group? And to make it more complicated, let's talk about glycogen targeting protein. That's the regulatory subunit of PP1. And it serves as a scaffold for the glycogen phosphorylase kinase, glycogen phosphorylase, and glycogen synthase. So you can think about it as, it is a docking point for all of these enzymes. So they're all together and they can all be phosphorylated or dephosphorylated quickly so that you can propagate a signal, the correct signal. Imagine if you had to go out and find all of the individual glycogen phosphorylase molecules and all the glycogen synthase molecules and dephosphorylate them all. Well, that would be a lot harder, and it would probably take more enzyme, and it would make the response less smooth and probably slow it down. But by having all of these things together with regulators, now you're cooking with gas. And it's a lot easier to have an enzyme come in and whoop, 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 do all the things. When you look at this figure, again, think about the hormone regulation, insulin, epinephrine, glucagon. Think about all the things that we've talked about and how it kind of fits in. So there are allosteric and hormonal signals that coordinate all of this carbohydrate metabolism. In the liver, we've got insulin activating glycogen synthase by inactivating GSK3 and activating the phosphorylase. We've also got glucagon that does the opposite. It stimulates glycogen breakdown and gluconeogenesis to generate free glucose, and it blocks glycolysis because we don't want to be burning up that glucose that we need to raise our blood sugar. In the muscle, we could talk about epinephrine and how that provides ATP by stimulating glycogen breakdown and glycolysis. So here's a figure that summarizes these things. You should be able to talk your way through it. And there's some mention of the regulation that we talked about in chapter 14 with hexokinases and PFK, pyruvate kinase. So this is a pretty rich figure that you can take time to pick apart and make sure that you understand how all of these things regulate one another. So let's talk about the differences in regulation of carbohydrate metabolism in muscle. So skeletal muscle, it uses its glycogen store only for its own needs. It's not shipping that glucose out to the blood. It is using glycolysis to make more energy. Skeletal muscle undergoes really big changes in its demand for ATP. So if you're just sitting down, listening to a lecture, taking notes, you're not really doing a whole lot. But then somebody comes in and scares you. Maybe you scream, maybe you jump to your feet and run, right? So that's a pretty big change in demand. And the muscle doesn't really have the enzymatic machinery to do gluconeogenesis, to make more glucose. And think about it, gluconeogenesis is a very energetically expensive process. If you need energy and you're already depleted and you need to very quickly go, you don't want to be doing gluconeogenesis. You don't want to invest energy like that. You just need more glucose, period. So think about that in terms of, how the skeletal muscle works to liberate its own glucose from its glycogen stores and why that makes sense. Carbohydrate and lipid metabolism are integrated by different regulation mechanisms. So the metabolism of fats and fatty acids is very closely tied to that of carbohydrates. There are some linkers like pyruvate and acetyl-CoA. And the regulatory controls adjust the metabolite flow through all these various pathways without causing huge changes in the concentrations of the intermediates shared with other pathways. So you're keeping a constant concentration of these metabolites by hormonal and allosteric mechanisms. So it's pretty crazy, all of the regulation that happens and how one molecule can literally regulate 3 different enzymes just to coordinate all of these pathways so that you're not, spinning your wheels or, spinning in circles, not getting anywhere. You don't want to defeat yourself metabolically, right? That's it for chapter 15. Short and sweet. Make sure that you pair this information with chapter 14. These two really do go hand in hand. So make sure that you do the work to kind of bring together all that hormonal regulation and have a good understanding of it. And remember to think about the biochemistry of it all. We're going to bring that in class, especially looking at some of these enzymes like the phosphorylase in class and doing some case studies. So thanks for watching. Be safe.